am Rachel Nethery. I'm a faculty member in the Department of Biostatistics at the Harvard School of Public Health and one of the CAFE Research Coordinating Center uh, com- uh, uh, team members. And I'm delighted to welcome you to this October edition of the CAFE University webinar series. These webinars are intended to be educational in nature, and they cover various climate and health topics presented by occasionally CAFE internal folks and typically visiting experts and lecturers from around the world. And content varies from data science related tutorials, grant writing tools and tips, and lectures on advanced topics within climate and health. And all of the webinars are made freely available on the CAFE YouTube site afterwards. So today we're extremely fortunate to be joined by Chris Golden, Laura Elsler, and Jessica Zamborain Mason from the Harvard Chan School of Public Health. Chris is an associate professor of nutrition and planetary health there, and Laura and Jessica are research associates working with Chris's team. Their team has developed a really cool collaborative research program to evaluate connections among climate change, fisheries management, and ocean governance, and food security and human nutrition in coastal populations around the world. And today they're going to be teaching us about how to harness data science to understand the nutritional importance of aquatic foods to healthy and sustainable diets. And so we're actually going to start off this presentation with a brief recording from Chris, and then we'll hand it over to Laura and Jessica to lead the latter parts of the session. So I believe, Elaine, we are ready for you to share the recording.
Hi, everyone. I'm excited to open up this data science training for us today with a very brief presentation on the importance of aquatic foods to broader food systems and to human nutrition and the climate threats to their sustainability. Here's our greatest challenge. We're now living on a planet threatened by forest loss, climate change and extreme heat, soil degradation, nutrient overloads in our aquatic systems, and mass freshwater overuse. Each of these environmental trajectories threatens our ability to produce food. Now let me explain two of our greatest health challenges that really represent two faces of the same coin. On the one hand, we're not eating the right types of food, leading to very high rates of inadequate micronutrient intakes. And this paper that was published a few weeks ago by my team highlights the risks to billions of people to micronutrient deficiencies given these inadequate intakes. On the other hand, the proliferation of Western diets and transitions to eating energy-dense, processed foods, sugar-sweetened beverages, large amounts of animal-sourced foods, etc., we're starting to see an increase in obesity, cardiovascular disease, type 2 diabetes, hypertension, and certain types of cancer. These two diet-related issues, inadequate micronutrient intakes on the one hand, and diet-related diseases driven by overconsumption on the other hand, are often existing within the same country and need to be simultaneously addressed. And this is why the Eat Lancet Commission on Healthy and Sustainable Diets has proposed the Planetary Health Diet to guide us toward healthy bodies and a healthy planet. This diet is plant forward. It includes animal-sourced foods, though, in kind of extreme moderation, and it especially encourages aquatic animal-sourced foods. And the real importance of animal source foods is really not to do with the delivery of protein, but it provides high quality micronutrients like iron, zinc, calcium, vitamins, and long chain fatty acids. And to understand kind of which foods provide these high concentrations of these nutrients, we needed a new database. And that's something that Laura will dive into soon. But when we looked at these vital nutrients, we found that the highest concentrations were observed in aquatic foods. And those pictured at the top were on average kind of the most nutrient rich. Those pictured on the bottom, so everything is ranked vertically here, were the least nutrient rich. And so you can see that the top 7 most nutrient rich across the suite of micronutrients that we looked at were all blue foods or aquatic foods. So aquatic foods are a great source of nutrients for people, but aquatic food systems are facing threats from unsustainable industrial fishing and climate change, a topic that Jessica will be diving into after Laura goes over the database. So the IPCC models are really looking at the impact of climate change on fish catch, and particularly looking at increases in sea temperature and how this will force fisheries to migrate polewards. And you can see in the dark red the areas highlighting where fish catch will be reduced the most. And so from a public health perspective, you're seeing the very areas highlighted where food insecurity and nutrition is the worst, where we struggle with marine governance, where there very well could be a kind of looming food crisis driven by this reduction in seafood attributed to climate change. And this is a threat to food systems and nutritionally vulnerable nations globally, because as you can see here, some countries are obtaining up to 100% of their nutrient requirements just from their small-scale seafood production. And so what our team did, this paper is now almost 8 years old now, I guess. Our team overlaid these climate models mentioned before and predicted the nutritional vulnerability of every coastal nation on Earth, looking at where they were currently consuming, which foods they were currently consuming, and how much that nutrient supply would be reduced from an associated reduction in fish catch driven by sea temperature rise. And what you see highlighted in orange are the countries that have the highest seafood dependency and are at a threshold for nutrient deficiency. And the areas in blue in the exclusive economic zones or in the ocean are the areas that are most at threat from fish catch declines driven by sea temperature rise. So where those two areas overlap, you can see kind of West and Central Africa and Southeast Asia are the areas that are likely to have this kind of looming food crisis that I mentioned. And so what do we do about this? Our team just published this paper a few weeks ago showing the benefits that could be derived from marine conservation efforts. And if we implement marine protected areas and rehabilitate fish stocks, we can increase fish biomass and therefore increase fish catch and fish consumption. In the upper right quadrant, you can see those countries that are darker in red are the populations that could have the greatest impact with the implementation of MPAs, marine protected areas, where you have a larger population, you have a higher estimated biomass impact from the implementation of those marine protected areas, and you can serve the most people because they are currently kind of inadequately nourished. Each of these papers that I mentioned have relied on our aquatic food composition database and data science approaches to harnessing this nutrient composition data. So Laura will now present on the aquatic food composition database, and Jessica will be presenting on data science applications to understand the climate change implications on aquatic food systems and on human nutrition. Thank you very much.
Okay. So we will now be turning it over to Laura and Jessica.
Okay, I start sharing my screen. Thanks so much, Chris, for the handover. We've been already getting quite some nice insights on the aquatic food composition database. The flagship paper Chris has shown you before has been published in 2021. That published the first version also of the AFCD. I'm just going to continue referring to the database as AFCD from now on. The idea behind the database was to acknowledge the importance of aquatic foods in comparison to all other foods regarding nutrition security and food security, but also to understand the different nutrient profiles, especially micronutrient profiles that vary a lot in comparison to, for example, just looking at proteins alone. The database has been compiled from food composition tables and more than 1,600 peer-reviewed papers that have published on individual species or a whole suite of different aquatic taxa and different nutrients. The figure that Chris has already shown you gives you a bit of an idea on the different types of nutrients that are included, from long chain fatty acids all the way to minerals and vitamins. And we can compare those to the recommended nutrient intakes to then understand how relevant or to which extent can they fulfill human nutritional needs, and can compare them also across land-based and ocean-based species. So, the database has more than 370,000 individual observations across 37 variables. And just giving you a couple of the selected variables, those include the scientific names of species across different taxonomic levels, all the way up to kingdom, and then different taxonomic codes, such as the ISCAP codes, FAO codes, then the and the Sea Around Us database. And then there's also information about the food itself. So has the food that has been studied in the individual, either coming from a report or food database, had it been dried, had it been boiled, steamed, all of those interact with micronutrients and the nutrient concentration of the individual species. As we're preparing them, we're also changing their nutrient composition. Then which food part had been used? Is it muscles, bones, or other parts of the food? Which literature, which was the study type, so the source that we've been using, the edible proportion, different types of nutrients, the nutrient codes and nutrient units. There are more than 200 nutrients included in the database, and some of them have a much higher sample size, like proteins, for example, and some are much less frequently reported on, such as fatty acids. There are 2,800 individual species that are included, and you can see at the bottom, most prominently, finfish has the largest sample size and is most frequently included in those nutrition studies. So there's quite a bit of empty spaces across different when looking at other classes. Where we see there are still really large gaps also regarding collection of micronutrient data. Then there's different food parts that I've already discussed a little bit before. So it's possible to have, you could eat an animal whole, or just eat the muscle tissue, which is most frequently just a filleted version of fish, for example, that is most often also reported on. But also oil, head, gills are in different cultural settings or in different ways of preparation, important components of aquatic species that have come with different micronutrient concentrations. Because we have these different components of aquatic animals that are more eaten or less eaten, or not possible to eat as well, the edible proportion ranges from 1, the entire animal, all the way down to 0.05, where you could imagine more crustaceans, for example, mollusks, which are often just a small inner part that's edible. And I wanted to bring you a little bit into a coding example to get a bit of a more hands-on understanding of the AFC database. There's a library. I'm going to give you a little bit of a, I'm going to show you where to find the AFCD later on. But so we can pull in the library, we can install and then pull in the library. And here I'm just including a set of nutrients more for our visual understanding and for example purposes here. And also I'm pulling in or I'm creating here a small data set with dietary reference intakes. So that's the that is how much does a single human need in a day to be consuming of those micronutrients. All right, I'm just going to switch you over into my R. All right. And so once we've been already loading into the database, as I've been telling you before, and we've been we can go through different you know, possible cleanings. You might want to change your nutrient names, which I have done now, going from AFCD to AFCD clean. And I'm going to group all the data by the scientific name of the species, the food part, the way it's been prepared, the product category, which I'll show you in a minute, and the nutrients, and summarize the values. So taking across those 5 categories, taking the mean values of each of the nutrients. And the result of that I want to show you for a moment. So here we have the scientific name of the species. We have the food part. So this could, for example, be the muscle tissue. The food preparation has been raw. So we could imagine a sushi. It's been wild caught, which is the product category. And we're looking here at the nutrient calcium at a value of 26 microgram. So we're going back. What I want to be looking at is this very simple example now of 2 species that have, you know, take different parts in our food system. The one is Salmo salar, commonly known as salmons, also one of the most, you know, commonly used species or most commonly consumed species. And the second one is the Pacific sardine. The Pacific sardine is often wild caught and used in fish meal that is then fed into aquaculture. So you might find the Pacific sardine also on your plate, but often it's also used as fish meal. And so then the fish meal is used to feed salmon. And one of the questions that I've been asking in the past year is, are we using those species efficiently? So would we have a better micronutrient benefit if we would directly consume Pacific sardines compared to feeding them to salmons and then eating the salmon? I am pivoting the nutrients just to be next to each other in the table, selecting for those species. And because for salmons and the specific question that I've been asking, we're mostly interested in farmed, I'm going to select for the farmed species here. I'll just take a few of the nutrients now inside and divide them by their recommended intake levels. And provide you now a bar plot on the different on the ratio of the nutrient content across each of those micronutrients for salmonids and for sardines. And the result is, I'm just going to make this result larger and pull you in there. Sorry, Elaine, can you give me a quick feedback if we're able to see the figure?
Yes, we are.
Yeah, okay, perfect. Yeah, so this here is now the comparison of the different nutrients. And we have iodine, iron, vitamin E, vitamin B12, calcium, zinc. All of those are important micronutrients specifically from aquatic foods. And in comparison to the recommended intakes, we can see, sorry, this is a little bit in the way because you cannot see. The Salmo salar is here represented in blue, the Pacific sardines in yellow. And across this specific set of species, the two in comparison here, we can see that sardines tend to have a higher nutrient concentration in relation to the recommended intakes than salmon. So this is a very simple example, but it can give us a first indication of showing whether a system, for example, that would output salmons for direct human consumption, but feed in Pacific sardines to produce those salmons, if that's an efficient system for improved micronutrient consumption amongst humans. Going out of this very small example, I'll bring you back into the presentation. All right. So the first database version has been published on the Harvard Dataverse. You can download it from there, and it corresponds to the 2021 paper. And the second version we're hoping to publish by the end of the year on GitHub as a package, as the way I showed you in the presentation and in the short excursion into R. And you will be able to find it on the link provided here in this presentation. There's until the end of the year, we're still hoping to finish a couple of improvements, such as updating existing values and adding additional studies. One, for example, around seaweeds and invertebrates, where I've shown you before that there were quite some gaps. Then adding accurate spatial and temporal data on where the samples had been collected, because the same species does not have the same micronutrient composition depending on where it's located and when it has been collected, for example, depending on the seasons. Then differentiating across different sample preparations and processing methods. So a deep fried fish and chips might not have the same micronutrient values as we would find, for example, in a raw version, such as sushi and sashimi. Then better accounting for potential duplicates as they're from peer-reviewed literature, maybe already taken into some food composition databases, and so on. And then finally creating and releasing the R package to make the data accessible and transparent. And sorry, I'm switching into Jessica's slide, but I'm going to stop sharing and hand over to Jessica for some more content.
Hello, good morning, everyone. So let me share my screen right now. Can you see my screen? Yes. Well, thank you, Chris and Laura, for that great introduction, and Rachel and Elaine for hosting us. So for the remaining of our talk, I'm going to give you a brief overview on how we're using the aquatic food composition database to answer questions related to climate and health. Yes. But before diving too deep into that, I wanted to give you a brief overview of the range of topics that our lab is working on using AFCD, which I'm also going to call it like that from now on. And this goes from examining how expanding marine protected areas around the globe can improve human nutrition, like Chris touched base on, but looking also at the efficiency and equity of aquaculture that Laura also touched base on. But we're also working on other projects like developing nutrition sensitive fisheries targets, looking at how thermal stress impacts or is likely to impact nutrient supplies, and I'll dive more into that in the next step. But on top of that, we're looking at other things like developing predictive models of nutrient content for invertebrate fisheries. As Laura mentioned, we don't have good representation of invertebrates and other taxonomic groups. So lots of the work we're doing is trying to predict nutrient content based on, for example, life history traits. We're looking at how aquatic foods contribute to achieving nutritional adequacies, doing this at global and local scales, examining the nutrient potential of seaweed, quantifying how, for example, artificial coral reefs can, how the implementation of artificial coral reefs can help people's nutrition security. We have some projects examining the trade-offs between nutrient supplies, but also heavy metal toxicity, and others looking into the spatial-temporal variability in spatial-temporal variability in nutrient content in aquatic foods, how processing, for example, affects nutrient quality and the policy implications of that, the nutritional selectivity of fishing gears, and then also looking even at the social and demographic determinants of nutrient catch profiles. And we do this at global scales, but also at small scales, for example, in Kiribati, Madagascar, and Papua New Guinea. But in today's talk, I am going to talk to you about a global study that uses the aquatic food composition database to look at thermal stress. And more specifically, the coral reef fish biomass and nutrient availability losses from thermal stress. And this is a paper that's currently in review, but the preprint is available and accessible for anyone wanting to have a deeper dive. As Chris mentioned earlier, aquatic foods provide critical nutrients to human populations. And this is especially the case for tropical coastal reef regions. And a clear example of that importance and the importance of reef fish for nutrition is this photo from Peter, a young man I had the privilege to meet in Papua New Guinea. In his hand, he has a reef fish that his mother caught that exact morning on the reef. If Peter's mother hadn't caught that fish, Peter would probably only have rice or another staple that day. And here is a broader example with data on how aquatic foods contribute nutritionally to people's consumption, this time from a representative national survey of food intake in Kiribati. On the y-axis here, you have different nutrients, and the grids are color-coded by the percent contribution of different food groups on the x-axis to household intake, on average across the entire population. And the food groups on the x-axis are all by contributing most on average across nutrients. So high contributions from a food to a given nutrient are in bright colors, and low contributions to household consumption are in darker colors. And we are finding that aquatic food consumption is the main contributor of key nutrients, providing, for example, about 50% of household vitamin B12, heme iron, retinol, niacin, vitamin A, and protein, which are critical nutrients for human health. And if we look at specific aquatic food groups, we see that reef fish in particular is the main contributor to household nutrient intakes under this study context. Now, this figure that I've just put up is the same, but now the x-axis are different seafood groups. So you have reef fish first, then you have pelagic fish, invertebrates, tinned seafood, dried and salted seafood, salts and raisins, seaweed. And what I really want you to take from here is that reef fish is really critical for these coastal tropical regions. However, simultaneously, tropical coral reefs are one of the most impacted ecosystems in the world from climate change. And here you have an example from American Samoa. On the left is a reef before a major thermal stress event, and on the left, is the reef before the big thermal stress event, and on the right you have the coral split after the thermal stress event impacted that ecosystem. As you probably know, the Earth is experiencing rapid warming, and this graph is a clear example of that. On the y-axis, you have temperature anomalies, and on the x-axis, you have different years. And we can see that, especially in the last decades, temperatures are exceeding those from historical baselines. That is true also for our oceans. And here you have the temperature anomalies on the y-axis and time on the x-axis. And you can see that in the last decades, there has been a rapid warming of our oceans as well. And this has impacted all ecosystems, but for example, on coral reefs, we are seeing altered ecosystems directly from increased thermal stress that lead to bleaching, like I showed in that figure, or the figure you have on this graph too, but also indirectly through increased sea level rise, increased frequency of destructive events such as cyclones, that also lead to increased runoff and sedimentation on reefs, altered ocean currents that decrease or change the connectivity among reef systems, which are quite patchy, and increased ocean acidification that affects especially those calcifying organisms like corals, but also others like invertebrates. If we look at thermal stress alone, we see drastic impacts on coral reef ecosystems. And for example, this is the consequences of one single big thermal stress event on the Great Barrier Reef that took place in 2016. And it was estimated that with that single thermal stress event, around 30% of corals, which are the foundations of coral reef ecosystems, died. Now, here you have the figure, and that's the Great Barrier Reef. And it's color-coded by the estimated change in coral cover from that event. And in red showing that some locations lost around 60% of coral cover from that big bleaching event. And then in green, some locations, especially towards the south, that had slight increases and weren't as affected. Unfortunately, though, these bleaching and thermal stress events are happening worldwide and more frequently and more intensively throughout the years. And here is another study looking at percent bleaching in different reef sites. So there's a different points are color-coded by that. But what I really want you to take from here is that it's not just on the Great Barrier Reef, but it's happening in all biogeographical regions. Increased thermal stress affects corals, the foundations of coral reef systems, but it also has direct and indirect effects on other parts of the ecosystem, such as fish assemblages, and also other ecosystem services like fisheries, shoreline protection, but ultimately can impact nutrition, which is what we're really going to talk about today. And here's an example from the Seychelles on how thermal stress can impact, for example, reef fish assemblages. On the x-axis here, you have different reef fish groups which are functionally important for the reef ecosystem. And on the y-axis, you have the relative contribution of those groups to overall reef fish biomass. In Seychelles, reefs were monitored before a big bleaching event and after that big thermal stress event. In blue, you have assemblages from sites that were recovering after that thermal stress event and were still in a coral-dominated state. But in red, you have assemblages from sites that had shifted from a coral-dominated state to a macroalgae-dominated state. And what I want you to take from here really is that after big thermal stress events, reef assemblages can change, as in this example. And this, as I briefly mentioned before, can ultimately affect the fish resources that are available for harvest and fisheries, and ultimately the fish that are contributing to people's nutrition, like we saw with Peter's photo before. Sadly, our oceans are expected to continue warming in the future, and this is the global sea surface temperature on the y-axis and the expected temperature throughout the century and the different climatic scenarios, or representative concentration pathways. These are the different emission scenarios that are projected throughout the future. We have a fossil fuel scenario in red and a pathway with climate mitigation, for example, in blue. And we see that we do expect, regardless of the scenario, an increase of sea surface temperature warming, especially more exacerbated under more greenhouse gas emissions. And these projected increases in temperature are expected to affect corals themselves. And here, for example, you have the number of global coral reefs that are expected to experience annual severe bleaching events throughout the century. And what I want you to take here, really, you have both scenarios, but what I want you to take here is that with increased global warming, almost all reefs on the planet are expected to experience annual severe coral bleaching. And with mitigation scenarios, so lower emission scenarios, making that happen at a later state. Future increases in temperature are also expected to negatively affect overall marine biomass. And as you can see in this graph, with more projected losses under more emissions. So here on the y-axis, you have the change in expected change in biomass as a percentage. And on the x-axis, you have time, and you just have different projections under a more fossil fueled development pathway, which is the red, and then under mitigation measures, or a middle of the road scenario in blue. However, what we really wanted to ask is how could this projected thermal stress impact reef fish assemblages and ultimately the nutrient supplies that are available for coastal populations? And to answer this question, we merged species-specific nutrient concentration estimates from the Aquatic Food Composition database with species-specific coral reef fish data from underwater surveys for more than 1,500 coral reefs around the globe. And we then, for these sites, for each of these 1,500 sites, we extracted historic yearly remote sense thermal stress estimates, which is measured as degree heat in weeks, which is a very common measure used for coral reefs, and also extracted other factors known to impact reef fish assemblages, such as the level of human impact. And we combined all this information to actually estimate the cumulative impact of thermal stress on total reef fish biomass, so the overall biomass that is on the reef, the biomass of different trophic groups or functional groups, as I showed before in the example from the Seychelles, and the nutrient availability of 14 nutrients that are considered essential for human health, especially in tropical regions. And then we used this estimated effect using that empirical data and extrapolated it to future thermal stress projections on all coral reefs around the globe. And this was done to estimate or give us a scenario of how future projected thermal stress could impact reef fish biomass and nutrient availability. And ultimately, we merged those projections with national nutrient inadequacy estimates to locate geographically reef locations that may be prioritized for future nutrition interventions. So going to the results, first I want to show you the data that we used to build the model that then we use to make inferences with future projections. So here is a map of all coral reefs, which is the grid in light, I would say beige or light orange that you can see at the back. That is all coral reefs around the globe. And on top of that, you have points which are our sampled sites. And dots are color-coded by the mean thermal stress experienced in that 10-year time span, previous 10-year span since they were sampled. And the size of the dots is scaled according to the maximum thermal stress that those sites experienced in that 10-year time frame. And it's more to see, okay, to acknowledge our biases on the data that we have at hand and what are the ranges of thermal stress that they have experienced, and especially as we're projected in the future, to acknowledge them. And here's the estimated cumulative effect size once we account for other confounders. And we have for total biomass in blue, we have for the biomass of different trophic groups in green, and the nutrient availability of different nutrients in purple, but similar to what Laura showed before, they're represented as human requirements to be able to compare across nutrients. On the y-axis, you have the expected cumulative percent change in biomass or nutrient availability from thermal stress with a unit increase throughout the decade. So if every year there was a unit increase throughout the last 10 years, if that makes sense. And on the x-axis, you have our different response variables. The bars are the median estimated effect sizes, and the lines are 50% uncertainty intervals from our Bayesian models. And in other words, this shows us that how, given available data, how we expect biomass and nutrient availability to change with increased thermal stress. There are really two key messages I want you to take from this figure. The first is that even with some uncertainty, we find that the cumulative effect size is negative for all response variables. So reefs that have experienced greater thermal stress in the past have less biomass of all trophic groups, and less nutrient availability. The second message I want you to take is that thermal stress does not affect all species groups equally. If overall biomass, for example, is expected to decrease around 20%, with a unit increase in thermal stress, the biomass of other groups like herbivores, which are fish that eat on algae, are expected to decrease much less, and I think it's around 5%. And others like macroinvertebrates, which are fish that feed on invertebrates, are expected to decrease a lot more, like around 20%, I think. And it is similar for nutrients, although with less magnitude differences, with nutrients such as vitamin A expected to decrease more with thermal stress. I think it was around 19%, than others like iodine, which are expected to decrease on average around 12% with a unit increase in thermal stress. And really what this shows is that thermal stress affects species differently, and as Laura showed before, species vary in their nutrient concentrations. So that is what our current state-of-the-art data tells us. So what if those relationships are applicable throughout the future? Here you have the thermal stress projections throughout the century for all coral reef sites in the world and the different climatic scenarios. In dark is a shared socioeconomic pathway dominated by high green gas emissions, and it's called the fossil fuel development pathway. And as a dotted line or in lighter colors, you have the expectations under a scenario with substantive climate mitigation. And it's specifically a scenario that's called middle of the road that assumes that warming is going to be restricted to 3 degrees Celsius by 2100. And the lines in this case are medians expected thermal stress across all reefs, and the polygons just show the ranges. And obviously we expect greater thermal stress to impact coral reefs in the business, in the fossil fuel development scenario. If we get our estimated effect sizes and project potential biomass changes, this is what we get. So in blue is the median expected change in overall biomass of reef fish across all reefs, and other colors are just different biomass groups, herbivores in green and macroinvertebrates in orange. And the dotted line, similar to before, is under a climate mitigation scenario, and the solid line is under a high fossil fuel development scenario. And just as a reference, the horizontal line is 0, so it would mean no change. And what you can see here is the average across all reefs. We see increased thermal stress is expected to cause overall reef fish biomass to decrease monotonically throughout the century, regardless of the climatic scenario. And however, within that, some groups like herbivores are expected to first slightly increase and then decrease, somehow consistent with those single case studies that we were seeing from the Seychelles. And those slight increases in trophic groups, however, do not compensate for overall biomass losses. And we expect with all nutrients on reefs expected to decrease monotonically throughout time based on our thermal stress relationships and on thermal stress projections. If we compare though both climatic scenarios, so if we subtract one minus the other, so the fossil fuel development scenario and versus the middle of the road scenario, we can get the prevented losses from climate mitigation throughout the century, which has potentially strong policy implications. And it shows that on average across all reefs around the globe, reaching that middle of the road shared socioeconomic pathway, besides allowing reef assemblages more time to potentially adapt, can have substantial medium-term benefits to human nutrition. Now, while up to now I have shown median expected changes across all reefs, I think it is critical to highlight that there is substantial spatial variability in expected changes given future projections in thermal stress. And here's a biomass example. You have sampled reefs in panel A and all reefs in panel B. For sampled reefs, we have the actual biomass on reefs on the y-axis and the expected changes in biomass throughout the century on the x-axis. And this is the marginal effect only from thermal stress. And this, I know this figure has a lot of information, so please bear with me as I go through it. So the dark line is the median expected change in biomass across all sampled reefs. Solid line is under a high emission scenario, and the dotted line is under a climate mitigation scenario. And the blue lines are individual sites under different scenarios. And what I really want you to see here is that there is substantial variability in expected changes, even if those median conditions show high declines. The dashed horizontal line represents 100 kilograms per hectare, which is a common threshold for coral reef fisheries to indicate a status of fishery collapse. Typically, when a site or a reef passes that threshold, we tend to say it's in a status of collapse. And the density plots above the figure show the year that each of those different sampled sites is expected to reach that threshold value. The dark density, again, is based on that high emission scenario, which shows that, for example, by 1100, around 98% of the sampled reefs are expected to reach that threshold based only on thermal stress. And the lighter color is under a middle of the road scenario that shows that by 2100, by 2100, 83% of the reef sites sampled are also expected to pass that threshold. But I think importantly, those density plots show the high variability from site to site. If we look at coral reefs worldwide, we can also see big differences in the expected changes in reef fish biomass by the end of the century. And this is, I have plotted here under a climate mitigation scenario. We have the same for the high emission scenario, but it just becomes a lot darker. And in this case, dark blue colors show greater expected changes per reef area, and lighter colors show less declines per reef area. Interestingly, though, if we group these grids that we have for global coral reefs, and we group them by jurisdictions, which are places like locations that have their own exclusive economic zones or ozone areas, we could also say that. We see which locations, based on current inferences and projections, are expected to have the highest declines in reef fish biomass, both in terms of total losses, given the country's reef area, which is panel C, but also losses per reef area, which is currently panel D. So here's panel C, and you have the rank of jurisdictions based on their expected total biomass loss by 2100 under a high fossil fuel pathway. And on the x-axis, you have reef area. Low ranks, such as 1, in this case, I think it's Myanmar, means that the country ranked the first in terms of total biomass losses. And points are individual jurisdictions, and they're color-coded by the total mitigation benefits. So in terms of how much reef area they have, how many kilograms of fish could they gain by implementing that middle of the road scenario versus the high fossil fuel development scenario. So in other words, lighter colors indicate which countries would gain the most from climate mitigation. And as you can see here, jurisdictions that have the largest reef areas like Australia, Thailand, and Myanmar are expected to lose the most reef fish biomass overall from increased thermal stress. And not surprisingly, given their extensive reef areas, are also the ones that are expected to gain the most from climate mitigation. However, if we look at changes per reef hectare per se, instead of total losses that account for the reef area, different jurisdictions pop up as losing the most biomass per reef area. And here you have the ranking of countries based on mean biomass loss on the y-axis, and you still have reef area on the x-axis. And now jurisdictions are color-coded by the mean biomass mitigation benefits that they could gain by implementing climate mitigation as opposed to that fossil fuel development scenario. And basically what we see by doing this mean instead of total losses, we see that there are other jurisdictions like Israel or Jordan that are expected to lose the most reef fish biomass per reef area, and other countries like United Arab Emirates and Bahrain that are expected to gain the most by the end of the century from meeting lower emission goals. Now, if we get these country-specific losses now in terms of nutrient availability, which is most of what our lab is really interested in, and we match them to jurisdiction-specific nutrient inadequacy intakes, we can see which regions that are already at risk of nutrient deficiencies are also expected to lose the most from reef fish nutrients based on thermal stress, and therefore which regions may be prioritized for future research and nutrition interventions based on the current data that's available. So here you have a map, and the reefs are now color-coded in like a purple color, and they're coded by the expected change in nutrient availability per reef area, doing an average across all the 14 nutrients that we examined. And the countries in this map are color-coded by the percent of the population estimated to have inadequate intake. So also average across nutrients that have that data available. So in other words, reefs that are darker purple are projected to lose more nutrients from reef fish by the end of the century, and countries that are darker are expected to be currently at higher risk of nutritional deficiencies. And if we do that for individual nutrients, we can, oh, I think I skipped one part potentially. If we do, yes, so we can do that combined as I showed in that graph, but we can also do it for specific nutrients, like we have here the case of vitamin E and the case of vitamin B1. So now here on the x-axis, we have the inadequate intake, so the percent of population having inadequate intake of that specific nutrient, in this case vitamin E or vitamin B1, and on the x-axis you have the ranking projected total losses of that specific nutrient for that country, and on the bottom right corner basically you're going to have the countries that are simultaneously currently at high risk of nutrient deficiency and that are also projected to have high losses from increased thermal stress. For example, in the case of vitamin E, we have Hawaii as high risk of vitamin E deficiency, which is categorized by that 50% of the population estimated to have inadequate intake of vitamin E, and it's also expected to lose substantial nutrient supplies from coral reefs because it's ranked among the top ones of the ones that have inadequate intake for that nutrient. And basically, we can do this for different nutrients, and we can locate which countries rank both high for having high risk of deficiency, and also rank high as potentially having high losses in their coral reef fish nutrients. And this individually can help inform future nutrition interventions, and it can also add the nutrition dimension of the relevance of climate policy, particularly for tropical nations. So, okay, so I wanted to finish this part of the talk giving some future research avenues that could build upon our work to increase our understanding of reef futures and ultimately the impact of climate change on people's nutrition, particularly for people like Peter that depend highly on tropical coral reef systems. So the first is to examine spatiotemporal variability in thermal stress association. So here we use this global data set covering 1,500 coral reef sites, but we use a space for time substitution to infer thermal stress associations. And even if that's the best of the data that we have at hand, there might be specific locations, for example, that have different species compositions that likely differ in how they respond to thermal stress. So our thermal stress models could be applied to places that do have temporal dynamics as these become available on reefs to inform specific regions and also to explore additional potential reef fish biomass and nutrient availability futures. A second priority is to further sample, as we're already working on AFCD, and as Laura mentioned, but it's to further sample reef fish for their nutritional content and examine, for example, how thermal stress also impacts nutrient concentrations in reef fish. We have some ongoing work trying to detangle the spatiotemporal variability in nutrient concentrations and how potentially thermal stress could impact that, but we're still, it's work that is still in progress. So adding that spatiotemporal variability component in nutrient concentrations within species will further inform future nutrient projections on coral reefs. Having better data, for example, not only on nutrient composition, but also on other factors like coral complexity or coral cover of specific reef sites, besides thermal stress, which is a remotely sensed product, will also help us discern the direct and indirect effects of thermal stress on reef fish assemblages. For example, it will help us tease apart how does habitat-mediated effects like bleaching affect reef fish assemblages and nutrient supplies in comparison to, for example, direct thermal stress effects on reef fish per se through increased temperature and its effect on metabolism. What we were able to do is like a mix based on the data we have, but having that extra information could discern the relative importance of those factors. And ultimately, I want to emphasize that thermal stress is one factor affecting reef fish assemblages, and that's what we were wanting to tease apart, but it's not the only one. For example, others such as the level of human impact that reefs experience have very strong associations, while we try to account for it to isolate the effect of thermal stress. Ultimately, if we really want to project the future nutrient changes that people like Peter are likely to experience, we need to account for that cumulative effects of all those factors affecting reef fish assemblages, fisheries, and ultimately the food people have access to. Okay, so I'm going to wrap up with a few take-home messages from today's talk. The first is that AFCD was created to acknowledge that diversity of nutrient profiles of different aquatic foods and the importance of aquatic foods in enhancing current and future nutrition security and public health. Our team is continuously improving AFCD, as Laura showed. There's lots of work in progress to create that version 2.0. And interestingly, AFCD can be applied in multiple ways, and I just showed you one way that we're applying it. Our team is developing a range of projects using AFCD to increase our understanding on the interconnections between aquatic resources, fisheries, aquaculture, human health, and especially in the context of climate change. But there is a lot more to be done. So if you're interested in this space, please stay tuned, and we're here if we can help answer any questions. Before I wrap up for the Q&A, I wanted to say that everything that's been presented here would not be possible without a big and evolving team. So thank everyone for that. And with that, I will probably stop sharing and jump to you for Q&As.
Wonderful. Thank you so much, Jessica, Laura, and Chris. This was really, really interesting, and I think a topic that a lot of people in the climate and health field don't necessarily know that much about. So this was really, I learned a lot from this experience. There are a handful of questions in the chat, and I will just open them up to whomever wants to speak up and answer them. So first, are we seeing any evidence of adaptation by the ecosystems to the impacts of climate change, or is the change that's happening, such as, you know, in these bleaching events or with the temperature changes or things like that, is that happening too rapidly for adaptation to really, you know, occur?
I can jump in for that. So I think it's a mix of both. I do think there are systems that are adapting, especially specific species, I would say. But I also think that if you zoom out and look at the overall picture, yes, warming is happening too fast to give a chance to assemblages and especially ecosystems to adapt to that. It's specifically to our study, we did we did do a sensitivity analysis, putting some changing the allowing the baseline to be adapted, like the baseline that we used to define thermal stress, to allow it to increase little by little, assuming assemblages could potentially adapt to that gradual increase in warming and not the big fluctuations. And we were still seeing monotonic decreases, at least monotonic decreases in biomass were expected. That said, I think we need to know more and look at specific locations that do have temporal data to really discern what assemblages are more robust and how that could impact human nutrition.
Yeah, maybe I can just add quickly to what Jessica said. I think the point that you also had in the presentation, Jessica, that there's many different types of stresses, really reduces the resilience of different species to so one species might be better to adapt to thermal stress, but not so well for other types of stressors, such as loss in nutrients or loss in oxygen. So as we're seeing all of those changes in the ocean happening at the same time, we're also seeing an overall global decrease in biomass that is coming along, but with really high variability. Some species that are more traveling to poleward, there's a lot of poleward migration happening. But of course, there's also some other taxa that are just much more benthic and wouldn't be able to migrate as much. So those are typically especially more vulnerable to pulse events.
That makes sense. Thank you both for your insight on that. Another question from the chat. Do the corals come back after they are bleached? And if so, how long does it take?
So yes and no. I'm going to give mixed. So when a coral is bleached, it doesn't necessarily mean it's dead. It's just expelled. It lives in symbiont with some microalgae, and the thermal stress just makes the coral expel those symbiont which are providing the coral with most of its food. Now, if thermal stress is prolonged, yes, ultimately that coral starves to death, and because it doesn't have its main food source. But in other instances, when thermal stress is slightly controlled, the coral is capable of reabsorbing, let's call it, its symbionts and not die. So I think in terms of time frame, it really depends on how strong thermal stress is.
Great, thank you. Let's see. Another question, have there been any policy changes to either limit confounders or additional stress on the corals, or to help the local population maintain fisheries?
Can you repeat, sorry, or have there been any policy change to it? Yeah, okay. Yes, I think it's very location specific. So I know there's been lots of, for example, water quality is a big one that interacts with thermal stress to affect coral bleaching and in different ways. So we're talking back to those cumulative effects. So I do think there are local and factors that can help mitigate to some extent the impacts that we observe. But overall, we need strong climate policies to really attack the causing problem, which is like fossil fuel development, high green gas emissions that are really affecting corals around the globe. So you can have increase a little bit the resilience in specific locations by not having that cumulative effect, but eventually if thermal stress keeps increasing, there might not be enough local or regional management measures that you can adopt to mitigate that.
That's a really good point. I actually have a follow-up question to that. So obviously, yes, ceasing fossil fuel emissions and all of those very large scale things would have tremendous impact. But what are some of the like local or regional things that you have seen implemented that do have an impact? So obviously, it may not be as significant of one as say, ceasing fossil fuel emissions, but have you seen specific, on a local or regional level policies or implementations that have had a positive impact?
So yes, I would say yes, with an asterisk, knowing that broader problem. But I do think, for example, there's this cumulative range of impacts: overfishing, low, poor water quality that are affecting reef systems as a whole. And maybe if you restrict, for example, if you have good fisheries management, effective fisheries management in place, you can still support human nutrition through species assemblages that might be changing in response to climate change. They're still changing because we're not stopping that big problem, but we can use these other effective fisheries management, effective good water quality that can enhance the system that's there, even if it's slowly transitioning to an alternative stable state because of thermal stress.
Maybe
Laura.
Mention additionally, because one of the projects in our team as well is around artificial coral reefs, which is one possibility that's much more local in scope to address climate change impacts on aquatic foods. And for policies, I think every country, it's interesting to look at every country has commitments to the UNFCCC that are both in terms of mitigation and adaptation, and they have increasingly included much more ocean-based policies. So it's a trend in the past 20 years, and I think the conscience is also increasing that aquatic foods need to be part of that system.
Wonderful, thank you. It looks like we have one more question in the chat. Is thermal stress measured relative to usual temperatures in a given location?
So in our case, we use degree heating weeks, which does use a baseline. I think it's from like what most climate models use. I think it's from it's a range in before the big increase in temperature from fossil fuel development. So we do use that baseline to categorize whether a place is experiencing thermal stress or not. We don't use different baselines for different locations, if that's the question.
Okay. Yes, that makes sense. Okay. I don't think we have any more questions in the chat. Again, I want to thank the three of you for joining us today. This has been a really fascinating talk, and I can't wait to share it with our community. Of course, the recording for this webinar is going to be made available on our website and on our YouTube soon. So be looking for that. And again, I just want to thank you, Jessica, Laura, and Chris, for joining us today. And yeah, I hope that we can expand upon introducing this work to the field in greater capacity, and really appreciate you spending the time with us this morning.
Thank you for hosting us.
Thank you so much, Elaine.
All right. Thank you, everyone. Have a great day.